So we're into the urinary system, which on a certain level looks pretty simple. It looks somewhat like the respiratory system in that it's a very compact system, relatively few organs. So coming off the digestive system, maybe it's a little bit of a relief. But it's a little deceiving in that way because, as we will find out, rather than being just solid tissue as the kidneys look from the outer surface, in fact they're extremely complex when you look on them, when you look within them. It's a series of all kinds of tubes, a million of these functional units that are called nephrons that are constantly working on the blood, conditioning it, regulating the content, regulating the volume, things like that. So let's take a look at this. So like we said, this is a relatively, from the surface, simplistic system where we have two major organs, the kidneys. We have the ureters that drain them. And then we have the bladder where we collect that urine, and then a urethra that drains that urine to the outside. So of these four major organs, what we'll find is we're going to spend most of the time on the kidneys because that's where all the work is done. For these other organs of the urinary system, ureters, bladder, urethra, it's just about storage and transport. So, we will have to learn a bit about those, but for the most part, right here is where we're going to spend our time and effort. Alright, so even when we start to look in on the kidneys, we can see that their internal anatomy is pretty complex. We see a series of blood vessels going throughout them, and notice that they're kind of working their way towards the outside. So we have a cortex out here, we have a medulla down here, and these blood vessels are working their way towards the very outside into that cortex, and that's where a lot of the work is going to be done. So when we see this vascular system, it's not only to keep the kidney itself alive as an organ, but it's also to deliver blood to the organ because, like I mentioned, the kidney has a job to do on the blood itself. So just like we saw with the liver, for instance, where it had additional blood vessels in order to exert some sort of effect on the blood, the kidneys are certainly doing that. And so if we were to look at the distribution of blood to the different organs, the kidneys get a disproportionately large amount, again, because they have this work to do on the blood itself. Okay. So this kind of gives you an idea of that flow pattern where we're going from our largest arteries to our largest veins into and out of the kidney. You're going to see renal all over the place. That refers to kidney. But notice, here's a unique aspect of this. We already have an idea about how the cardiovascular system works, arteries to arterioles to capillaries to venules to veins. But here we're seeing something of that, right? So you're seeing this flow pattern. But as we continue around here, we get to capillaries. Okay, good. We recognize that. It comes after arterioles. But look at this. It flows into an arteriole, and then there's a second set of capillaries as well before we start going to venules and veins. So it's a little bit different that way, so we're going to look at why that might be. Alright, so when we get down, after you get down to the macroscopic anatomy, you really look inside of the kidney, we get down to the workhorse of the kidney, which is the nephron. So you need to know all about the anatomy and the physiology of the nephron. What are the different components of the nephron and why we have each one. So when we talk about the glomerulus and the glomerular capsule, when we talk about the proximal convoluted tubule, this windy tubule that we see here coming right after the glomerulus, why do we have that? We have this interesting looking what's called nephron loop that extends down into the medulla. We then get back up into the cortex and it starts winding around again. And then ultimately, at the end of that nephron, it's going to dump into a big tree-like structure that's called a collecting tubule or a collecting duct. But notice, we see that there are different sections of it. Just like the intestines, just like the digestive tract, we're going to see differences in terms of structure and function. You can even see it on a cellular level. So you need to know the parts of the nephron and what each part is doing. So we start at the beginning of the nephron here, where we see the interaction of two sets of tubes inside of the kidney. So we have vessels, right? We have blood vessels. So for instance, here's an afferent arteriole. Here's a glomerulus, this ball of capillaries right here. And then there's an efferent arteriole. But notice that here are the capillaries. That's where we get exchange occurring. Now, what we were seeing for previous systemic capillaries is that it was a true exchange. Things were moving out, things were moving back in. We learned the forces involved. We talked about hydrostatic pressure or blood pressure. We talked about osmotic pressure and things like that. But here, in this ball of capillaries, we do get movement across what we would call the filtration membrane, but that movement is always going to be out. And so we're going to see fluids and small solutes leaving the capillaries and being collected by this surrounding glomerular capsule. And then this fluid and these solutes are going to go down a different pathway, a different set of tubes, which are going to be contained within the rest of that nephron, right? Proximal convoluted tubule, loop of Henle or the nephron loop, and then distal convoluted tubule, etc. So we see a separation. And that, as this name indicates, is what's called filtration. And filtration occurs in the glomerulus. Okay. So we also notice as we're looking at the anatomy of the nephron that there are two different types. There are cortical nephrons, of which most of your nephrons, you have 1 million per kidney. Most of them are cortical, occurring mostly in the cortex. There are some that are juxtamedullary, that go just that much further into the medulla, like we see down here. This is medulla down here. So we'll talk about why you have those two. But again, regardless of whether they're short or long renal nephrons, what we find is that there's going to be this interaction between the tubules themselves and the cardiovascular system. So again, here's that glomerulus, the ball of capillaries, like you see here, an afferent arteriole coming in, an efferent arteriole coming out, and then we also have this peritubular capillary bed. So this is going to be another site of exchange with the tubules of the nephron. So two sets of capillary beds, each one where substances are passing across the wall. So we go back to the glomerulus. We're starting there as the beginning of our nephron. We talked about the filtration that occurs there. Here you can see a zoomed up view of that proximal convoluted tubule where the fluid's going to be moving out. And what we find is that not only do we need to understand the structure of how these parts of the kidney are doing their job to produce urine, but we'll find every time when we're talking about these new systems, we need to understand how this system is controlled. How do we increase its activity? How do we decrease it? And as we were seeing with digestive system, etc., and cardiovascular system, there's usually an intrinsic level of control where it says, I'm fine, I can control this, I can handle things on my own. And then there's extrinsic control by the nervous system and by the endocrine system that are coordinating it and overseeing the activity that's going on inside of the kidney in this case. Okay, so we see one of those control points or checkpoints here called the juxtaglomerular complex. It's also called the juxtaglomerular apparatus. So this is just next to the glomerulus, and it's a series of cells that regulate the filtration that is occurring. Now, it also will regulate other aspects of the blood concentration and blood content because we have sets of cells here that have different jobs to do. So here it says juxtaglomerular complex, and what we find is that there are going to be specialized cells of, usually we call it the distal convoluted tubule, but sometimes they say it's just the last section of the ascending limb of that nephron loop. We see that there are cells right adjacent to it, where we call these extraglomerular mesangial cells, and there are also glomerular mesangial cells like we see here. And then, see how this afferent arteriole has a thicker wall to it right here? These granular cells that we see here, or juxtaglomerular cells, these ones also have a job to do in terms of helping to, like we said, control filtration or regulate the content of the urine that we're producing. So we'll get into the details of that as well. That's a separate kind of small topic where, again, the kidneys and these small nephrons are able to adjust and control the work that they're doing. Okay, so when we do this filtration, which is the first step in this process of creating urine, we're going to be moving things across this filtration membrane. We said that it's kind of unique because when we're moving things across here, what we're going to see by all my arrows is that the movement is always out of these glomerular capillaries. There is a significantly higher blood pressure inside of the glomerulus because if we look at the diameter here of the afferent arteriole going in compared to the efferent arteriole going out that's draining it, we can see there's a much larger diameter or much more blood flow coming in compared to going out. So what that does is that creates a backup. It creates an increase in pressure here, which then pushes fluids across this filtration membrane. And that big hydrostatic pressure or blood pressure that we see here, it's so much larger than the regular capillaries. It overcomes any of those other different pressures that we talked about creating exchange at the systemic capillaries. So in terms of osmotic pressure and the hydrostatic pressure that's trying to push back the other way from the fluid in the glomerular capsule, it cannot overcome this net outward pressure and this net outward movement that we're going to get. Okay. So what we've been talking about so far, we've been looking at this here, the beginning of the nephron and this process here, which is called glomerular filtration. So glomerular filtration occurs right there at the glomerulus. And then we have two other processes as well, though. Tubular reabsorption and tubular secretion. And these occur in the simplified diagram. They occur in the rest of the nephron. So the proximal convoluted tubule and the nephron loop and the distal convoluted tubule as well. And even a little bit in that collecting tubule. And so if you look at it, right, you can see that reabsorption is where we're taking solutes and water from the tubes and we're putting them back into the cardiovascular system. Right? We absorb these things first in the digestive system. Now we are almost going to lose them because if we were to follow this out, right, we would be peeing these structures out, or the solvents, etc., and solutes out as urine. So if we want to get them back, we want to reabsorb them, we've got to pull them out and put them back into the blood. So that's reabsorption. Secretion is movement the other way. This is where there are additional substances that we want to get rid of from the blood, and we're going to push them into the filtrate. Okay? So those three processes are key. You need to know those three processes, be able to define them, be able to show where they occur, and give examples of what type of substances are moving according to these three processes. So, for instance, water gets filtered, actually water gets reabsorbed, sodium does get filtered as well because it's small, sodium does get reabsorbed, things like that. Okay, so it goes through a little bit of glomerular filtration. This is just showing us with the size of these arrows, the forces involved here. And again, you can see how the outward moving forces definitely overcome those smaller inward forces that are trying to go back the other way. So we get a net outward movement here of our solutes and our solvent, basically water. We talked a little bit about how the kidneys work to control themselves when we were talking about that juxtaglomerular complex. But then we do go through the mechanisms that influence the activities of the kidney in terms of neural controls as well as hormonal controls. As we progress our way down, it's kind of like the digestive system. We start with the beginning, as you do, talk about what's happening at the beginning of this system and of this microscopic workhorse, the nephron, and then we pretend that we're urine working our way down. What's going to happen as we reach these other segments? And we said that reabsorption and secretion are these other two processes. Both of them are important. So we talked about reabsorption, what it means, and what things are pulled across and how they are pulled across. So this process is highly selective and highly variable. It means we only pull certain things back, and the amount that we pull back will vary depending on the body's needs. So for instance, water. If you're drinking a lot of water, you certainly do not need to hold on to more. In fact, you need to get rid of that excess water so that your blood volume and blood pressure doesn't go up too much. So even with something as important as water, the amount of water that we reabsorb or let go is going to be variable. Now, while we're talking about this reabsorption of certain substances, many of them can reabsorb ad nauseum. We just pull them back as soon as we need it, and it's not a problem. But in some cases, we'll find that for particular solutes, there can be this thing that's called a transport maximum as well. This isn't a real centralized topic. It's not going to be one of our larger topics of the chapter, but it is something that we address. So again, we talked about how these different solutes and solvents are brought back. The water that we reabsorb, we actually have two types of water reabsorption. Obligatory, where we have to pull back a certain amount of water so that we don't filter out all of our plasma and then just be a dried up husk. And then there's what's called facultative water reabsorption, where the remaining amount, about 20%, we have a choice as to whether we need to get it back or whether we should really get rid of it. Okay, so just different types of reabsorption going on. Then we move on to secretion. We understand these are substances moving the other way, being pushed into the tubules from the capillaries. And we just need to understand what types of things we'd be pulling back. So for secretion, the most common things are going to be potassium ions and hydrogen ions. The last major topic that we have in this system is what the heck is that loop of Henle and what does it do? And the main role of this loop of Henle is that it creates what's called a vertical osmotic gradient in terms of the concentration of the tissues of the kidney. You can see it goes 4 times as concentrated down here in the deeper parts of the medulla compared to the cortex. And the main thing that that does is it allows us, if necessary, to concentrate the fluid that's contained within these tubules. So here's that loop of Henle. That loop of Henle actually creates this vertical osmotic gradient, and then in this last tubular segment here, in the collecting tubule, if we need to reabsorb water, then we can use that gradient, use the osmotic gradient to create osmosis and reabsorb water from the tubule back into the cardiovascular system. So that's if you're really dehydrated, this osmotic gradient allows us to reabsorb that water as necessary. So you can produce, with this nephron, like we said, this gradient that we see here. And what that does is in this last tubular segment, in the collecting duct, it allows us to reabsorb water as necessary. Now this is called facultative reabsorption because, again, it's only if we need to will we reabsorb that water. So if we reabsorb that water, that extra water, then we produce a very high concentration but low volume urine. If we don't need to do that, then basically we just close the doors on this process, and then all of that water that was starting in the collecting duct ends up leaving the tube as well, in what we would call dilute urine. Okay, so there are some summary figures you'll find at the end of the chapter, which are very important in giving you an idea about, again, what the different parts of the nephron are and what is occurring in those parts. So you should know things like this, right? So you see the arrows coming this way. These arrows are showing you reabsorption. We're pulling it from the tubule and we're putting it back into the blood. The arrows that point into the tubule, that's secretion. So we're actually secreting these things into the filtrate to get them out of the body as urine. So you can see proximal and distal convoluted tubules have a lot going on, as well that last collecting duct or collecting tubule. And as I mentioned, the rest of the urinary system, we can't disregard it, but the ureters and the bladder and the urethra are pretty simple structures. So as long as we know the anatomy, the physiology is so simple, it's an easy thing to tack on to the end there. But the main place that we're focusing for urinary system is at that nephron and understanding what it's doing inside of the kidney to produce urine.